Bismillahirrahmanirrahim. Now this is the second video on the March 22 paper 1-2, which is on the new syllabus, and everybody must go through this video before you take the exams on the 9th of June. Now starting with question number 21, some parts of a typical are more numerous than the others. Which parts are listed in order from the most numerous to the least numerous? Centromere, nucleotide, histone. Now, first of all, we've got to understand one chromosome is made up of one DNA molecule. It's made up of two strands, but that is one DNA molecule. It's two strands, but it's one DNA molecule. In every chromosome, there are two telomeres. In every chromosome, there's one centromere. So this is what you have to understand is how you're going to be deciding this. Two telomeres, one centromere, one DNA molecule. Now the answer to this was C. Why was it C? Because there are many histone proteins. There are these blocks of 8 histone proteins around which the DNA is, there's a double helix is wound around it, just like you have a thread around a reel. So histone, telomere, and then DNA molecule. One DNA molecule per chromosome. Question 22. Which row correctly describes a stage of mitosis? Now you got to understand the stage of mitosis is nuclear envelope. It's got to be clear. Now here it says reforms, begin to form, not present, disappears. Now this word is wrong. Nuclear envelope disintegrates. It doesn't disappear. It doesn't something doesn't disappear. There's no such thing as disappear in biology. So disappear is the wrong word, and that's why you would have gone for A, because it does disintegrate in the prophase. So the answer to question number 22 is D. Why is it D? Because telophase, nucleus envelope reforms at a maximum distance from the cell equator, and spindle microtubules break down. You see, the confusing one was A, in which you could have made a mistake in that, but the word disappears is wrong. So that is why you see these papers need a lot of time for you to read and to reconsider your answer. Question number 23. The diagram represents a nucleotide containing adenine. So the base here is adenine. Which statement about this nucleotide are correct? The carbohydrate is a pentose. The base contains nitrogen. The base pairing, if this is adenine, it can either base pair with thymine or adenine can base pair with uracil, but this would be in the RNA. But they have not said anything. They have not specified in the question whether they're talking of DNA or RNA. They're saying, so yes, the base pairing can occur with uracil. Now, adenine is a pyrimidine. No, I'm sorry. Adenine and guanine are purines. Now, this is something that we have to remember. It's purines. You have to remember this by some mnemonic, that adenine and guanine are purines. So, that is why this was wrong. So, part 4 is wrong. And the rest of them are correct. So, the answer to 23 was B. Then we say which row is correct for the replication of a DNA molecule. Which row is correct for the replication of a DNA molecule? So direction of DNA synthesis is always 5-3. So these two can be correct. 3 to 5 is wrong. We should know this. This is a basic fact. It's always in the 5-3 direction. Enzymes that add complementary nucleotides to the lagging strand. Now this is the new syllabus. So the lagging strand you've got to understand is that which is, you see when this opens on, one is continuous and one is discontinuous. That is the lagging strand. Now what adds nucleotides to the lagging strand? So you have to be correct on that. That was C. It's DNA polymerase. DNA ligase actually joins these Okazaki fragments. So DNA ligase joins these Okazaki fragments. Maybe you have a question on this in the coming exam. Now coming to question number 25, some students were asked to look at the photomicrograph of a cross section of unfamiliar material and describe what they could see. You see, here you get it wrong. What you could see, what you can see is what you are not going to interpret, and that is where you're going to get it wrong. The student described the cross section of F as circular hollow tube. How do you know it's a hollow tube? You don't see the 3 dimension. A hollow tube means it will have been something like this. Spherical means it will be round like a ball. We don't know. We've just seen a section of it. So the student described the section of F as circular, hollow tube, spherical. Which description of the cross section of F correctly state what the students could actually see? Could actually see You've got to understand is to actually see, you weren't seeing much. So the answer to this was C. What you actually see is C. Yeah, that's a bit of a joke. Circular. You could only see something which was circular here, but that's it. How do you know it's a hollow tube? How do you know it's round like a spherical means 3D round shape, which means like if I show you a cricket ball or a football, you would say, yes, that's a round spherical. So you couldn't see that. So please remember that, how you were going to interpret this. Now, question number 26, the diagram shows the relationship between phloem sieve tube element, xylem vessel, and companion cells. Which row correctly identifies what could be represented by numbers 1, 2, 3, 4, and 5? Now, 1, 2, 3, 4, and 5. Now, you have to look at these companion cells and phloem sieve tube. Is endoplasmic reticulum present in both? Phloem sieve tube element and xylem, no nucleus. Is that correct? So, always do it like this. You have to compare 1 and 3. And you have to see, is the 2 present in 1 and 3? Is the 4 present in 3 and 5? So as you can see, please pause the video here, read all this and then think what is the answer. And the answer is A. Why is it A? Because companion cells and phloem sieve tube have endoplasmic reticulum, and phloem sieve tube and xylem vessels do not have a nucleus. Now here is where you have to, companion cells and phloem sieve do not have the nucleus. So this was easy for you to pick up. And xylem vessel does not have cytoplasm, but phloem tube has cytoplasm. There is peripheral cytoplasm in it. If you knew that, only then you could do this question. Then coming on to question number 27, why does an air bubble in a xylem vessel element stop the flow of water? So this is the xylem vessel, right? And there's water flowing in it, right? And we have an air bubble in it. And this stops water from flowing. So water cannot go anywhere. So we have a problem. Now why isn't the water going? So it says, why does an air bubble in a xylem vessel stop the flow of water? Loss of adhesion, loss of cohesion, collapse of the xylem vessel element. Well, of course, 3 was wrong. It's not going to collapse it. But the water molecules have cohesive forces. So the water molecules' cohesive forces are lost. So that is why the answer to 27 was D, 2 only. Adhesion, so there's no question of adhesion because the water cannot go up. Then question number 28, which changes to the water potential and the volume of solution in a phloem sieve tube? Okay, let's imagine a phloem sieve tube. This again, we have a lot of problems with you imagining a phloem sieve tube. So this is a phloem sieve tube with the sieve plates here. Now, when amino acids are moved, so when amino acids are moved out of here, say amino acids move out, AA amino acids are moved out. So if amino acid moved out, what is going to happen to the water potential? So the solute decreases. So the water potential is going to become higher. When solute moves out, water potential becomes higher. So it could be A or B. But what will happen to the volume of the solution in the phloem sieve tube? Well, of course, because water is going to follow. So this is going to decrease. So this is how you have to do this, and the answer was A. So amino acids move out, water follows. Even if you just look at the solute moving out, water not following, even if you say that, even that would decrease the volume of solution in the phloem sieve tube. Now coming to question number 29, which feature of some xerophytic leaves reduces the rate of transpiration? Reduces the rate of transpiration by decreasing the water potential gradient between the internal leaf surface and the atmosphere. So we have a xerophytic leaf. Now the stomata are present here. Stomata present in grooves, sunken pits. Now when water molecules, water vapors, please understand this is not water, it's not a liquid form. When water vapors are going to move out, they're going to get trapped here. When they're going to get trapped here, well, there's going to be a decrease in the water potential gradient. outside there's less water vapor. But when these are going to be high, so then this rate of transpiration is going to decrease, because inside there are water vapors, and they're also in these pits there are water vapors. So that is why the answer to 29 was C, stomata located in sunken pits on the leaf surface. You see, all these others are not related to this. Thick waxy cuticle, yes, prevents water loss. The spines also less surface, so yes, elongated leaves, there are soreness. That means there's more water is stored. They are not in line with the question. They are not in line with the question. It says, how is the rate of transpiration reduced? Then coming on to question number 30, the diagram shows a section through a mammalian heart. Which chambers of the heart are represented by G and H? Now, I figured this out. As I looked at it, I said, oh my God, now this is some difficult question. I look at this and I said, okay, this wall is thicker and this wall is thinner. So I immediately thought this is something to do with the left side. So when you think of it, this is the left side, so this is the right side. And this is the left side, because it got a thick wall. Now, so which chambers, now it says which chambers of the heart are represented by G and H. So this had to be D, this would be the right ventricle and this would be the left ventricle, and these two things look like the bicuspid valve on the left side. So this is how you have to figure it out. Then coming on to question number 31. Now question number 31. Very interesting question. Blood entering the heart from the vena cava passes through or past several structures before entering the lungs. 5 of these structures are included in the list. Now, as soon as it enters, it passes through the SA node, sinoatrial node. This is the sinoatrial node here. Then it goes on to the AV node. So this is 1, this is 2, this is the AV node 2, and then the Purkinje tissue, which would be 3, which is this here. Then number 4 would be semilunar valve, and number 5 would be the pulmonary artery. So 5 of these structures are included in the list. SA node, AV node, Purkinje tissue, semilunar valve, which would be here when the pulmonary artery leaves it, then there's a semilunar valve here, and then the pulmonary artery which carries blood to the lungs, because that's what it said. Heart passes through or past several structures before entering the lungs. And then it says, after arranging these 5 structures in the correct order, blood flow from the vena cava to the lungs, which structure will be third? Purkinje tissue. So get this right. How did we say the 5 structures and how did we do this? So that is why I'm saying, you see, they've given you more time so that you can do these questions more profoundly, and you can think about it and you can spend more time on it. They've not increased the time just like that. Then question 32, the bar chart shows the quality of endothelial tissue, elastic tissue and muscle tissue in the walls of 3 blood vessels P, Q and R. So, I mean, first of all, let's look at P, Q and R. P has endothelial is the same in all of them. Then the muscular tissue, now this is the muscle tissue which is here. So muscle tissue is a little more here in Q and less here in R. Then the elastic tissue, let's color it another color. And the elastic tissue is this one. So there's more elastic tissue in P, there's less elastic tissue in Q, and there's very little elastic tissue in R. So one has to be very clear about what is going to be R. If it's got less elastic tissue and less, and the choices that you have is elastic artery, muscular artery, and vein. So there are the 3 choices that you have. So what you have to understand is R has to be the vein. So it can be either this or this. Now the answer is A, very simple. Why? Because the elastic artery, the muscular artery would of course be Q. It would have more muscle. And elastic artery would have naturally more elastic tissue. So the answer is A. This is of course again on the new syllabus and you must be knowing this. That is why you have to be clear on it. as to how this would be the different. They haven't given you the names of these vessels. I can think of you giving you an MCQ on the names of the vessels, because the vessels which are nearer the organ would have more muscles. So you must please read up on the muscular artery in the new book. Now, question 33. The maximum pressure in each of the four chambers of a healthy human heart was recorded during one cardiac cycle. The maximum pressure recorded were 3, 10, 25 millimeters of mercury, 120 millimeters of mercury. Now, I would try to fit these in. Now, I would know that this would be 120 millimeters. Why? Because this was the highest pressure which is generated in the left ventricle, because it has a very thick wall. It has to send the blood to the all around the body. And then you have a choice between 3 and 10 and 25. So I would say, well, okay, this would be, now they are asking you which value was recorded for the right ventricle. Now the 2, 3 and 10 would be more for the atria. Now, why the left atrium would be 3 millimeters of mercury? And why would this be 10 millimeters of mercury? And this would, of course, be 25 millimeters of mercury. Now, please understand why. The left atrium is receiving blood from the lungs. The pressure is greatly less, because in the pulmonary artery, the pressure is less. The pulmonary vein pressure is even lesser. Right atrium blood is coming from the superior and inferior vena cava. So there's slightly more pressure because the blood is entering from the body. And the two major blood vessels which are carrying the deoxygenated blood back to the right atrium. And then the right ventricle has a thinner wall. It has thicker. Left ventricle is much more thicker, but right ventricle is thinner, so it can generate some force. So that would be 25. So that is why the answer to this was C, right ventricle. What value was recorded for the right ventricle? Now coming to question number 34, the graph shows the pressure changes in three structures of the right side of the heart during the cardiac cycle. Right side of the heart. Now it says what is represented by the line labeled T. Now if you look at the one which is on the left side, this is the aorta, and the aorta is coming out of the left ventricle. So which one is the vessel which is coming out of the right ventricle? That is the pulmonary artery. So what you have to understand is that the answer to this was this: pressure changes in the pulmonary artery. If this was given of the left side, then you would say the aorta. But if it is given of the right side, then you know it is the pulmonary artery. But if you remember the cardiac cycle, only then I'm sure you can do this question. I'm sure many of you can do this question. Then coming on to question number 35, which statement helps to explain why there is no cartilage in the walls of the bronchioles? Cartilage would make the bronchioles too narrow. Gases must diffuse across the walls of the bronchiole. Rubbish. Smooth muscle is sufficient to support. You see, what is the function of the cartilage? Function of the cartilage is to support the trachea. Wherever there is, in the trachea, there's C-shaped cartilage. In the bronchioles, in the earlier bronchioles, there are plates of cartilage. The bronchiole wall do not need to change the shape. I mean, cartilage is not helpful in changing the shape of the bronchiole. So if you knew your syllabus, you know that the answer was C. Why? Because we have the smooth muscle to support the walls of the bronchiole. Basically, cartilage function is to support. And if you can understand that, then you can understand the essence of this MCQ. Question number 36, the photomicrograph shows a cross-section of part of the gas exchange system of a mammal. What is shown by the line labeled V? The diameter of an alveolus, the diameter of a bronchiole, the diameter of a capillary, the diameter of a trachea. Now, everybody should be clear. Trachea has a C-shaped cartilage. Capillary does not have anything in it. So either it's an alveolus, these are the alveoli. These are the alveoli. So you must know this. This is actually in your syllabus, the photomicrograph of alveoli of the gas exchange system. So that is why the answer to this was B, is the diameter of a bronchiole. Question number 37, which features of the disease cholera decreases the spread of the pathogen Vibrio cholera? Decreases the spread. So if somebody has cholera, what happens which will decrease the spread of the cholera? So number one, A, immunity to cholera after vaccination is short-lived, lasting less than 2 years after vaccination. This will of course increase the spread, not decrease the spread. Up to 98% of people infected are symptomless carriers. So this is even more dangerous. This will increase the spread. You have to understand, and one is the decrease, the other also have to be the increase. Cholera rapidly kills up 50% of people with symptoms if they are not treated. Well, that would be the end of it. Then it will decrease the spread. Simple rehydration therapy successfully treats about 99% of the people with symptoms of cholera. But that treats them, that doesn't decrease the spread of the pathogen. It treats them, but then they're still suffering from it. While they're suffering, they're still spreading it. So that is why the answer to this was C, because if they are not treated, then this will of course kill them. So this will decrease the spread of the pathogen. Question 38, which row shows how penicillin kills bacteria? Processes inhibited by penicillin effect on the bacteria. Now, what is the process that is inhibited by penicillin? Is the formation of the peptidoglycan crosslinks, breakdown of the crosslinks, formation of peptidoglycan crosslinks for breakdown. Does it inhibit the formation or the breakdown? This is the thing you've got to be clear. There are only two choices here. The process that is inhibited is either the formation or the breakdown. So are you clear about that? What does it inhibit? It inhibits the formation of the peptidoglycan crosslinks. And when this peptidoglycan becomes weak, it's not that strong anymore because these crosslinks are not formed, then what is going to happen? Water will enter and the bacteria will burst. So this is in reading the question that you have to be very slow and very careful and in understanding the question. What is the process inhibited? Is it the formation or the breakdown? If you know that, only then you can do this. Then coming on to question number 39. In the hybridoma method, what is grown by cell culture to produce monoclonal antibodies? Now, what were monoclonal? You took the B cells, then you took the myeloma cells, then what did you get? You got hybridoma cells. And these hybridoma cells will actually produce the monoclonal antibodies. So what were you doing? You were just using this cell, and what were you doing? You were cloning it. So you had taken the B cell and the cancer cell, and then you had fused it. You got a hybridoma cell. So in the hybridoma method, what is grown by cell culture to produce monoclonal antibodies? Is grown by cell culture is you've grown clones. So the cloning of these would result in the production of monoclonal. Of course, these will then produce monoclonal antibodies. Last question 40. Which type of immunity does a baby have at birth? Naturally, it's a natural passive immunity. At birth, what does he have? He or she has the ready-made antibodies, which the mother must have suffered from a disease, and then she's got those antibodies, and then she's just passing those antibodies on to the baby. So it's natural passive immunity. The baby's lymphocytes have not done any work. It's the mother's lymphocyte. She's had antigenic exposure. She's had B lymphocyte selection and cloning. Then she has plasma B cells. Then those plasma B cells have made antibodies. And those antibodies have been passed to the child. So it's passive immunity because antibodies have passed. And it's a natural process. We haven't done anything. It's not artificial. We haven't done anything in the lab. So please understand how you're going to handle the new MCQ paper. You're going to take your time. You're going to read the question very carefully. You're going to go over the question a number of times, and then you're going to think what is going to be the correct answer. It's a lot of thinking which you need to do. They want you to think more. That is why they've made the question slightly more difficult. Now, I wish you all the best, and thank you for watching, and thank you for subscribing. Thank you very much.